Hello and welcome to my channel. Today I'm going to talk about connection between granting and statistics and a waste of so many $1 million on pure noise. Let's begin. In depression research, many people who are working with depressed patients know already that there might be something like predisposition. There were even a notion that it runs in families because people who are working on that for quite a long time invested a lot of grit and persistence and hard work to find out what the causes of depression are. And still we cannot say that there is one simple thing you can point your finger at and resolve that and there is no depression. What I want to talk about today is one huge discovery in my eyes about completely wrongly funded 20 years of research. In 1996, Collier et al. And colleagues actually published a groundbreaking research about a gene which might be the cause of depression. That gene was called SLC6A4, and in 1996 DNA sequencing was not so cheap as is today, so they joined forces because this is a European study collecting a sample in Milan, London and Würzburg. And they focused on this particular gene because it is known that it has to do something with serotonin entrance into the cell. So they made an educated guess to put that as a hypothesis. As a result of their study, in next 20 years, there were more than 200 other studies studying exactly this, a connection of depression and genetic predisposition. It is trying to find one single gene which is the cause of depression. But it turned out to be those results are flawed. So they stated in their 1996 study that this particular gene, which they focus on, can be a source of detection of depression, cause of screening, even follow the progress of any kind of therapy, like finding out whether in certain person antidepressants work and so on. But none of them that showed to be true. How that can happen? The main result of Richard Boulder's and his colleagues' study from 2019 is that these genes have nothing to do with depression. Since we know much more about gene sequencing and the role some genes play and how actually they can impact something which is a far-stretch behavior, the truth is probably in the notion that there is no one gene for something, because many genes are working in collaboration, and for every single threat of a person or animal, you have to find several dozens or even hundreds of genes working together on something in order to have certain feature or evolutionary advantage so that organism survives. Well, this is a big question, among others, how to elucidate on antidepressants' effectivity, but I think I'm going to do entirely a new video about that. Let's say for the start that even so many years after Prozac was launched and used in so many people, we still don't know exact mechanism how that works or why it doesn't. So let's focus on new evidence. 2 years ago, just before this crisis hit, Richard Boulder from Colorado University Boulder actually published the study in 2019 after great comparative study of many basically volunteers who volunteered in gene sequencing. They proved that there is no evidence of genetic predisposition for depression. Basically, they compared many studies in the last 20 years who were trying to establish the connection between 18 different genes, which are so-called connected to this major one, which is a major player, SLC6A4. And in the end, they found no evidence whatsoever. Of that research, Matthew Keller actually stated, "We didn't find a smidge of evidence." He was also posing a question in their 2019 publication, how on earth it can happen that we wasted 20 years of work and hundreds of millions of dollars on non-existent evidence. They said that most probable the 1996 study was reporting on something that we called false positives. So how that can happen? Ioannidis published one study in 2005 showing that 65% of published data are simply not reliable because they're doing their statistics wrong. And this is a big controversy of so-called p-value mentioned in every single medical or psychological study. So there is a big movement to increase reproducibility, especially in psychology and other social sciences. But what I want to focus here is so-called PRISMA, which is basically a framework which encourages people to increase the feasibility and to increase the quality of the evidence that they are publishing. So I don't want to go in details because it is quite a complicated way to give people, how should I say, guidelines how to pre-register studies, how to in advance describe the methodology used, because there are many observed biases in the literature and in scientific studies due to mainly publish or perish culture everywhere, because people are forced to publish their data, publish their results, even if they are not actually useful in practice, because their job depends on that, their life depends on that, and people are simply finding creative ways how to publish something which is not publishable. There are a number of things that there is now initiative to correct for, and PRISMA initiative actually started after 2000, and simply when you go and look in the literature, do meta-analysis, you see that reliability of the published data is gradually increasing. This is not to say that people are doing this willingly, but for example, one of the reasons why study that I mentioned in the beginning is completely wrong, and people spent 20 years on studying basically noise, was so-called false negative. When you're doing hypothesis testing, you're actually finding evidence whether your hypothesis is reliable or not in evidence. What is the probability that your hypothesis is true? Contrary to non-existence evidence for that claim, which we call zero hypothesis. So the frequentionistic approach in statistics is actually trying to find whether there is an evidence that hypothesis is true. And basically, you have among your findings so-called error number 1 and error number 2. And error number 1 is so-called false positives. When you find there is an evidence for your hypothesis, when there is actually none. And in statistics, there are defined ways how you can correct for your type 1 error. But obviously, the authors from 1996 didn't do that. It is possible to detect the publication bias in biases, and there are ongoing culture of doing meta-analysis, because meta-analysis and this meta-analytic approach is giving us the chance to better estimate the data, or to find out what is closer to the truth from our data, the evidence which really has grounding in true effect. Next time that you're trying to start your study, there are many things you should do. you should first think about confidence interval, about the estimate of power, and you should look in the literature how to correct publication bias to avoid cherry picking, harking. This is one picture now. And also publication biases are not something that researchers are doing willingly, but in order to avoid that and to offer your fellow scientists and general population something which is practically useful, you should avoid those drops in statistics. Thank you for listening.